Hi everyone. My name is Dr. Chris Murphy. I am a lecturer in laser plasma physics at the University of York, and I'm also the admissions tutor here at Physics at York. Welcome to the first of our winter webinar series. It's great to have everybody here. So a little bit about the winter webinar series. We'll be bringing you cutting-edge research from the School of Physics, Engineering and Technology here at the University of York to give you a little glimpse into some of the range of exciting areas that our physicists and engineers are working on. As well as today's speaker, we have one other speaker before Christmas. That'll be Dr. Phil Hasnip, who'll be speaking about using quantum mechanics to design new materials. That's on Wednesday, the 11th of December. After Christmas, we'll have Professor Roddy Van speaking about fusion technology. We'll have Dr. Stuart Higgins talking about whether you can heal people with electricity. So it's a bit like pseudoscience to me, but apparently it's totally based on real sort of biomedical engineering stuff that we do here. We've got Dr. Edgar Buchanan-Berryman, who will be giving an insight into autonomous robot evolution. And the series will conclude with Dr. Mohammed Nasser Esfahani, who will be discussing engineering on the nanoscale to create devices and systems that are not only small, but also capable of unprecedented performance in fields like medicine, electronics, energy, and environmental protection. So a real broad range of science and engineering going on in this winter webinar series. Now to tonight's event, we are very lucky to have Dr. Agnes Noy joining us to give a talk. Agnes is originally from Barcelona, and she always wanted to understand how life works, which initially led her into biochemistry and chemistry, and then in computational chemistry, and then she realized that a lot of the interesting things that you need to understand about DNA comes from understanding the physics of the process. She continues her research in biophysics. She relocated to Oxford and then to Leeds, and she's started our research group here at York in 2016, where she's now a senior lecturer. So she's going to be talking tonight about her understanding of DNA and the research that she and her group do, about all the information that you can get, including some of the highest resolution of images, and showing really how a lot of these biomedical processes and biophysical medical processes are actually governed by the laws of physics, and we need physics techniques in order to understand a lot more about how DNA works, including the dancing of DNA inside our cells. I'm not going to pretend to know any more about this. I'm going to leave it to the expert, Agnes Noy, who's going to take it away and talk a little bit more about how she and her group do research into dancing DNA. Take it away, Agnes.
Well, so thanks for the introduction. So we are using physics here to understand biology, to understand how life works, and you will see an example of how physics can make a real important contribution on this. And so we have the image of DNA being a perfect double helix, a relaxed molecule, but this is not the reality. So DNA inside the cells suffer from mechanical stress. It's pull, it's bend, and it's twist. Yeah, so we are going to talk about this today. Now, okay, so just a brief recap of how the DNA molecule looks like, or what is it. So it's a very long molecule. It's a very long polymer made by a series of repetitive subunits. And these are the subunits. And each subunit, which is called a nucleotide, it has a part that it's always the same, is what we call the constant part, which is the phosphate and the sugar. And this makes what we call the sugar phosphate backbone. And then each of these repetitive units have a part that is different, and it's the base, and it can be 4 different molecules, and it can be an adenine, so we have an a thymine, we have a T, a cytosine, and a guanine, we have C and G. And then these really long molecules, these really long strands, come together, 2 strands together, and we say that they are complementary because the G binds to the C and the T binds to the A. Okay, so we have this polymer where the base pairs are making different levels, yes, and then each level stacks in top of each other and then make this molecule. And what happens here is the distance between one level or one base pair to the next one is shorter when we go through the base pairs and done through the backbone. So let me pointer options. Let me put the pointer options. So then to this interaction is really strong, it's really stable. The molecules, the bases really like to be in the top of each other. So then what happens to solve this difference in distance, and the DNA makes the helix. Yeah, so this is the trick that the DNA makes, the molecule makes, to solve this difference in the distance through the backbone and through the bases. So that's why the molecule is a double helix. Okay, so then what we have is that we have a very long molecule, and the order of these bases is what defines the genetic sequence, the DNA sequence, yes? And because this molecule is very long, we can have really long sequences. And then when we consider the whole DNA in the cells, we have the whole genome and we have the genome sequence, right? So these are probably concepts that you have here in the media and in books and all this stuff. So just to let us to make some numbers. So bacteria are much simple organisms. Their genome have 5 million bases. If we would extend the DNA just in a linear shape, it has a length of 1.5 millimeters and broadly 4,500 genes. If we have the human genomes, these are much more sophisticated genomes. They have a much more bases, 3 billion bases. If we would extend the DNA, it would be 1 meter long, and the number of genes, it's about around 20,500 genes. Okay, so the first thing that we realized here is that the length of the DNA is much longer than the dimensions of the cell. Yeah, the dimensions of the cell are in the microscopic wall, and this DNA, it makes millimeters or meters long. So the first physical problem that we have is that we need to compact this DNA inside the cells. Okay, the second one is that if, as we remember from the DNA sequence, the genetic information is inside the molecule, and the outside there is the backbone, which is always constant. So for the cells to read the genetic information, what it needs to do, it needs to separate the both strands of the DNA to create what we call a DNA bubble. And this is not a trivial problem from the physical point of view, yes, because here we have some forces, we are destabilizing the molecule, and we need to fight against some stabilizing forces. So here you have, let's imagine that this rod is my DNA model. So here we have the genetic information is kind of inside, and then we need to build this bubble, yes? Okay, so we have two physical problems to solve. So yes, so here just a brief kind of annotation for the reminder of the biology. Here what we have is the DNA bubble is formed. This genetic information is read by a protein that it's called the RNA polymerase, and then this is synthesizing the RNA. Then the RNA is just the messenger, yeah? Just the messenger that allow us to translate this information, this genetic information, to make the proteins, and the proteins are the molecules that does the job in the proteins, yes? So DNA does nothing apart of having the genetic information. RNA is just a messenger. The proteins, the molecules that do the work in the cells are the proteins, yes? And for example, this protein, what is its job is to read this DNA sequence, and it's called RNA polymerase. So also just a brief recap about DNA, RNA. They are very similar molecules, but they have two chemical differences, which are very subtle, very small, but the effects are really big. So the sugar here of the DNA and the RNA, so the RNA has an oxygen more, and then the bases are the same, except that the RNA has a uracil instead of a thymine. But if you look at the chemical difference, it's just this carbon. So you can see the difference are very little, but actually what happens is that RNA is more reactive. So it can react with chemically with lots of things. So RNA has a function sometimes of helping the protein. For example, it has been used as RNA vaccine. It's more active. While DNA is a very boring molecule, doesn't react with anything. It's just very stable because, of course, have the genetic information, it needs to be really stable and last for a long time. Yeah. Okay. But let's go back to our problem. How do we solve these two physical problems? Yes. The first is to compact the DNA inside the cell. The second is to separate the both strands of the DNA. Okay. So the trick that DNA, that cells does, is to untwist the DNA. So what happens here is that you will see here in my rat model, if you untwist the DNA, you can see that it becomes much more easy to separate the both strands of the DNA. Yeah, so you can do the bubble. The other effect is that if you might have experienced that using cables, yes? So if you untwist the DNA or over twist, that's okay. Let's untwist the DNA now. I'm just untwisting my rod. You will see that it creates these loops, these supercoil loops. So this is what happens in cells. So DNA is supercoiled. It makes these coiled coils, yes? So DNA is maintained in an undertwisted state, so it's more easy to separate the both strands, and also it makes these coiled coils, and then this helps to compact the DNA inside the cells. So this is an image of how the genome of a bacteria looks like, and you can see that this is just a bunch of loops, yes? So this is just a scheme diagram of that. So of course, animals and plants, we are much more sophisticated, so we need much more efficient ways to compact the DNA. And here the trick is to use these proteins, the histones, and where the DNA is wrapped around. But still the DNA is supercoiled in animals, plants, and us. Yep. Okay. So we have seen that so if we have the DNA untwisted, then this facilitates the reading of the genetic information. But however, the relationship between the reading the genetic information and supercoiling it goes in two directions, because here what happens is that let's imagine that we have our piece of DNA. I'm going to do this model with a rope, and here the protein, this RNA polymerase comes, yes, and start reading this, makes a bubble, a DNA bubble, and start reading this sequence, and then starts to move in one direction here. Yes, you can see what happens. So the DNA ahead of the protein, it's over twisted, and the DNA behind of the protein is under twisted. So the action of reading the DNA sequences also creates supercoiling. Okay, so you can see how this effect also helps to regulate the genes, helps to switch on or switch off the genes. So here we have an example. So for example, let's imagine that we have a gene. The gene 1 is here. So the promoter is just a small sequence that it's a sign for the beginning of a gene. Yes. So we have the promoter here, the promoter 1, which is the beginning of gene 1, and then the RNA polymerase have come here and has read this gene in that direction. So of course, the DNA behind the RNA polymerase is under twisted, and this then, if there is a promoter nearby, it will receive some undertwisting from the activity of gene 1, and so this activity of gene 1 will facilitate the activation of gene 2. Yes, so we have this this process of connection and regulations between genes that are relying on the physical properties of DNA. Okay. So, but how can we study the DNA? How can we see supercoiled DNA? So let's start with, so what happens so we know that DNA is a double helix because Rosalind Franklin used the X-ray diffraction, yes? So what she did is that she made a crystal of DNA, where all the molecules here, as you can see in this image, all the molecules of DNA are really well organized, are making a crystal, always in the same orientation and the same conformation. So then how this technique works is that then you have the crystal here and you irradiate an X-ray beam of light or radiation, let's say, and then this radiation is diffracted, is dispersed, and makes a diffraction pattern. And this is the famous image that Rosalind Franklin took. And from there, she deduced that DNA needed to be an helix. Yeah. And then Watson and Crick were the ones who did the actual chemical model, saying the sugar needs to be there, the phosphate and the bases needs to be inside. Yeah. So this is how we know that DNA is a double helix. And this technique, this X-ray diffraction technique, has been widely used to deduce the structure of all types of biomolecules. Yeah. So, for example, here it has been used to deduce the structure of the hemoglobin, which is this protein, and this is the protein responsible to carry the oxygen in the blood. So this technique has been widely used. However, it does not work in our problem for describing supercoiled DNA. And this is because supercoiled DNA, it's not always in the same shape. So sometimes it makes it look like this, sometimes it's slightly different. And so it's very bad for building a crystal. So here is when my research comes in. So what we did, together in collaboration with Alice Pine from the University of Sheffield, is combine microscopy and simulations. So Alice is an expert, a well-known expert in atomic force microscopy. And this technique consists in sticking the molecules in a surface. And then what you do is that you use a tip that scans the surface systematically and kind of fills the volume of the molecules that are stick there. And so by this filling this volume, you can deduce the shape of these molecules. So Alice has been improving this technique when it's used in biological molecules, and she has improved the resolution to a really good level. So she even could resolve the grooves of the DNA here. You can see these lines. However, as you can see, we don't have such a big resolution as the X-ray. Yeah? So in X-ray, we had a resolution of all atoms. Yes, we could deduce the coordinates of each atom. But here, using this technique, it's not possible. Yeah, but this technique, what it allows us is it allows us to describe flexible molecules and molecules and each one molecule at a time. Yeah, so if one molecule is slightly different than the other one, nothing happens because we are seeing the shape of each individual molecule. However, the resolution is not so good, but that's why we use computational simulations. Yeah, and here is what I did. So my expertise is running computational simulations on biomolecules, and these simulations are based in physical models. And what we do is that we calculate the force that each atom is feeling according to the chemical environment. And so, of course, if we kind of remind a little bit of the classical mechanics, if we know the force, then we can find out the acceleration. And if we find out the acceleration, we can find the velocity and then the coordinates after a time step. Yeah. So we do all these calculations over and over again for different time steps, for different time steps, so we can deduce the snapshots of how the molecule moves, how the coordinates change as a function of time. Okay. However, so we try to simplify these physical models as much as possible, because we have lots of atoms and DNA is very long, as we have seen, but still a kind of a simulation of this length of the DNA, which has around, I don't know, 100 or 100 base pairs, still needs a lot of computational power. So we use big computers to calculate these equations. And here, for example, we use the supercomputer, the biggest supercomputer in the UK, which is called Archer, is in Edinburgh, and it was inaugurated during COVID time. You can see here Boris Johnson, who was the prime minister at the time. Yeah. So with the use of these supercomputers, we can perform simulations on how the DNA moves. And also, another thing that I would like to say is that DNA moves because it has thermal motion, yes? In the same way as the molecules of gas moves in our room, yeah, in the air of our room. So the molecules in the liquids, they also move because they have this, they are in a particular temperature. But of course, the movement is not so big. They are not a gas because they are in a liquid, but still they move, right? So we need to take the image that all the molecules in our cells are vibrating and giggling all the time. Like for example, this kind of vibration, yeah? Okay, so we apply the two techniques to DNA circles that only contain 300 base pairs, okay? So you might say, well, I say only because 300 base pairs, it's not very big, it's not very long compared with a whole genome. But this system, this system of a study, it's enough to see what is happening to the DNA when the DNA is supercalled. Which was our objective. How the DNA, the supercalled DNA, looks like. So one of the things that we saw. So these are the images obtained by Alice, which are beautiful, and these are the images from my simulations. So one of the first thing that we saw is that when the DNA is undertwisted, it can present this variety of structures. So believe me, when the DNA was relaxed, it was just boring. It was just a circle of DNA, yeah? But when the DNA is supercoiled, it starts making all these shapes, right? And also what we saw is that actually our simulations could capture more or less the same shapes observed by the microscopy. So here the two techniques help each other, right? So through this comparison, we validate our simulations because our simulations are very beautiful, and very nice, but they are a model. They are not the truth. Yeah, they are not an experimental observation. And on the other hand, the AFM, they are very nice, but of course they are struggling with the resolution. So when the two techniques agree, the simulations kind of act as a boost of resolution from the experiment. So we could reproduce this, we observed this croissant shape here. We could also reproduce the triangle, the 8 shape here, the diamond, and what we call the hedgehog. So also what Alice did a series of snapshots after a certain time. So these are a series of time-resolved snapshots. So after every time she could capture a snapshot, and you can see here that the crossing of the loop is going up and down. So the system is dynamic, and I could capture the same dynamics in my simulations. So this is a series of snapshots. This is another series. So here I would like to show you a movie extracted from my simulations, and that is capturing the same dynamics that we observe here. And you can see how flexible, how dynamic is the DNA when it's undertwisted. So the DNA is not much flexible when the DNA is relaxed. So we said that the DNA is so flexible. Let's see if I can show the movie again. When the DNA is so flexible that actually we call it that it was dancing. It was just like dancing. And this only happens when it's undertwisted. So we published this paper. We were very happy about that. And we made a press release, a kind of article for the general public and called Dancing DNA. And we were very pleased to see that we received some media coverage. So this was covered by the Daily Mail and the Daily Express. And even Alice was interviewed in ITV. So yeah, so we were very happy to see that. And if you want to see this coverage, you can just Google dancing DNA, and you might see some of these articles. So another thing that we saw is that as we increase the torsional stress, as we under-twisted the DNA, one turn, two turns, or several turns, we start seeing these melting bubbles. Yeah. So these are the structures obtained from the simulations here in the right. So this is the DNA circle when it's relaxed. Here is when it's under-twisted, one turn, two turns, three turns, and six turns. And you can see how the double helix starts breaking and making these bubbles and also some kinks. So on the other, in the side of the AFM, so Alice could see that there were some discontinuities here in her images, but of course the resolution was not perfect. So then it was very useful to have the simulations to really confirm what she was seeing. Now, the other interesting thing that we see is that these bubbles appear between these two levels of of torsional stress, which is actually the level that is maintained in the cells. So, and this is interesting because the DNA can be transitioned between having less or more bubbles depending of the interest, the physiological interest, depending on whether it wants to be really active or more conservative. Okay, so we published this article. We were very happy, but one of the comments that we received, which is totally fair, is that, okay, you have observed these things on a model that it's a relatively small circle of DNA. How do you know that what you are observing is that it's happening in longer DNA or in a linear DNA, let's say? So we took this challenge in the simulations. So we have started modeling the simulations, the supercoiled DNA on longer DNA, and these are the movies. This is over-twisted DNA here. We have increased the twisting of the DNA, and this is under-twisted DNA. And the under-twisted DNA presents DNA bubbles here in red. And the over-twisted DNA does not present bubbles because if you over-twist the DNA, it's much more difficult to separate the both strands. So actually, the presence of the presence or absence of bubbles makes that the dynamics of the supercoiled DNA very different. Because here, what happens is that these bubbles are very flexible. So they act as a hinge, and they tend to be where the DNA is more bent, which is at the tip of this loop, at the tip of this coiled loop. Like when you kind of try to bend a pipe, no? So at the beginning, you try to bend a pipe, and then it makes a kind of regular curvature until you kind of break it, you kink it. And then that kink behaves as a hinge. Yes, that is a weak point of the pipe that kind of bends really easily. So the same happens to DNA. So let's observe let's go back one moment. So, okay, so we can see the movie of the over-twisted DNA. So the over-twisted DNA does not present bubbles, and what happens then is that it's much more dynamic. So there is no, at the tip of the plectonym, there is no bubble that it's spinning the plectonym. It's fixing the plectonym in a particular position, and you can see how this loop is kind of can move from the top to the bottom of the molecule, which is a thing that you cannot see in the negative supercoiling. So once there is a bubble, the bubble is at the tip of the plectonym. The DNA is so comfortable there in terms of energy, is not moving from there. Okay, so you might wonder why we are studying all this, apart of that it's beautiful and it makes these images so beautiful and help us to understand how DNA works. But what is the practical purpose to understand how DNA works? So just as a reflection, we are just making basic science on how of the details of how DNA makes the bubbles, how DNA organizes these loops, and you can see that these and this is setting the basis to make more elaborate models to predict when a gene is going to be switched on or is going to be switched off. And this has relevance, has an impact on bioengineering, because what is happening now is that we are starting to use organisms as biofactories that can produce products in our interest. Yeah? So, for example, biofuels or the synthesis of different molecules, food additives, medicines, beauty components, or for example, this burger, which does not have animal, it's not from an animal, it has been made by yeasts. Yeah. Also, we can use bacteria or yeast for cleaning the water. For that, these bacteria or these yeast can synthesize lots of proteins that can cut down or kind of clean the water. You know, this that can make some good chemical reactions. So this is also a way to do this much more environmentally friendly, because the organisms are making everything for you. That you don't need to use pure chemistry and use lots of water or some kind of molecules or reactants that are pollutant of they are not expensive or pollutant of the environment. So we are in the way of starting to use biology in our favor. And some of the progress have been done with the knowledge that we are having, but still, of course, we don't have yet a full understanding of how the physics of DNA is influencing the regulation of the genes. So we are expecting that once we can build more sophisticated models, we will be able to really understand how we can switch on switch up a gene of interest. Okay, so finally, the most important slide, which is the acknowledgements. So the simulations on linear DNA was done by Matthew, and then the simulations on the circular DNA were done by myself, but Victor helped me to analyze this. So Victor came from Mexico, and he brought a PhD scholarship from the Mexican government. Matthew, he was from the UK, and I also want to thank you, the PhD students, master students that have made my group. This is the people who is working at the interface between physics and biology in the physics department at York. So here you can see there are several academics, but also some researchers and also PhD students. And finally, this is Alice, who was my collaborator in Sheffield. So the funding agencies, I did my PhD in Barcelona, in Spain, and then I gained a fellowship from the Catalan government to came to the UK after my PhD to do my more research. So and then I established here, and I just want to thank the other funding agencies also from the UK. And thanks for your attention.
Great. Well, that was really interesting. Thanks so much for that, Agnes. Always interesting to hear about a topic that I know very little about. So it's always nice to hear a bit more of that. We've got a few of the attendees have asked a few questions, so I'll go through some of those questions. As I say, feel free to add more questions in the Q&A. We'll try and get through as many of them as we can. We've got about 20 minutes. We should get through many of these. Here's a question we've got. So you talked about the dancing DNA, and there was kind of almost a kind of Brownian motion argument I think you were making, that the sort of thermal energy that causes this. Is it random, or are there particular movements which recur? And I guess related to that, another person's asked, does the DNA not reach a position of equilibrium when it's over or under twisted? And I guess those two are related, maybe?
Yes. Okay. So what happens here is when the DNA is linear, it has a position of equilibrium, yes? So more or less it's being straight, and then it bends a little bit, makes that. When the DNA relaxes, it has a circle, the position of equilibrium is like this, and then it vibrates a little bit, but it's always around this structure. So, but when it's under twisted, what happens is that we are putting the DNA in an uncomfortable position, right? So the DNA has a stress, and it has a different ways to release this stress. And then, and then it is so the energy landscape becomes much more flat. So the DNA is much more what we call the physicists frustrated. So it can go to different positions, different states where to solve the problem that it has. So that's why it becomes so dynamic. So yes, it's random. So, but it always has. So it's random. It has a minimum, and this minimum, this shape depends on the if the DNA is in a stable or it's supercoiled.
So are there lots of kind of, I guess, local minima, and it can jump between them because they're quite shallow minima? Is that what?
Yeah, exactly. So, for example, when the DNA is supercoiled, it can do lots of things. So, for example, let's imagine that the DNA is supercoiled. It can make the loop like this, where the strong bending is here. It can also make the loop like this, where the strong bendings are in different positions. It can make a bubble, and then once they make a bubble, the stress is released to that, so the rest of the DNA can relax a little bit. So you can see that it's very dynamic because it has different ways that to go, or different postures that can adopt to solve this kind of problem that it has.
Yeah, and is DNA ever not supercoiled and therefore not dancing? I guess you kind of answered that.
Yes, so okay, so the cells tend to maintain a level of supercoiling there, just to keep the DNA active, because if not, it's difficult to separate the both strands in DNA and know what it tells. But sometimes, depending of the cells are spending energy to keep the DNA uncoiled and twisted. So depending of the levels of energy of the bacteria, happens in bacteria. It can increase the torsional stress or decrease the torsional stress. And for example, if it has lots of food, it's growing a lot, it can increase the torsional stress, so everything is going faster. No, all the metabolism is going faster. If it's kind of entering in a kind of it doesn't have food, or it's kind of entering in a relaxed or kind of sleeping state, then it relaxes the supercoiling. It has less energy. It relaxes supercoiling, so less things happen. So it's a way to turn off the DNA a little bit.
Excellent. Yeah, there's a question here, and I worry this might be kind of too far towards the medical applications, but I'll ask the question anyway. Um, how close are you to bridging the gap in a person that has a DNA mutation missing 3-7, like a DMD sufferer? So, like, I think that's Duchenne muscular dystrophy. So how close is this to understanding, I guess, DNA mutations and missing DNA? Is there medical applications which might be interesting for this?
Yeah. So yes. It's interesting. So let's imagine that we have so a mutation is when this process. So the DNA needs to. Let's say the mutation happens when something. I don't know. There is a kind of reaction in the DNA. No. So then. It's instead of having an AT, we have a TT, or we have what we call a mismatch, which is a wrong pairment of the bases, or GG, or what something it's not right, right? So then this double helix is imperfect. So then what happens is when we have a mutation of DNA, it's much more easy to make a bubble, yeah, to break this double helix structure of the DNA. So we have here a bubble or a kink, this kind of disruptions. And what happens is that this is a thing that is what we are learning now. It's quite new, but what is being seen is that these mutations are very flexible. They act like kings, and they tend to be at the tip of the plectonyms of these coiled lobes. And then a protein can go and recognize that, and they kind of set a flag to the cell saying, oh, this is here, there is something happening. And this activates the repair mechanisms. The cells have some repair mechanisms. So yeah, so this physical model is also important to understand mutations and how they are repaired.
That's really interesting. Yeah, there was a comment from this is a comment more than a question about the red and white cord. People love it. People love your demonstration. They say it's really interesting, really illuminating, really helps understanding it, which is excellent. Um, there's a question: what potential of energy is the DNA ultimately trying to reach? I think I'm not sure what that. I guess it's just lowering the potential energy. Is it? Is that?
So yes, it's a little bit what I said before. I'm not good. So when the DNA is relaxed, it has the natural propensity to twist. It's in the lower level of energy. As we twist the DNA, we are kind of making the DNA unstable because the DNA has the tendency to have this natural propensity to twist that is determined by these stacking interactions, this kind of one stacking the other. So this what is defined what defines what is the natural propensity of the DNA to twist, and if the DNA can be in that state, is when it's more comfortable. It's in the lowest state of energy. Something. So if when we under twist, over twist, we are kind of adding potential energy in kind of way.
Yeah. I've got another question here. Is the pairing effect of GCAT similar to enzymes? I guess.
Okay. Okay, so let's see if I can understand that. So yes, okay. So I think so the enzymes have the product. So okay, I understand. I think I understand the question. So when the the genetic information is read, so what happens is that there is a protein that detects that there is an A, for example, there, and then puts a T. And if there is a C, he puts a G, and then if there is I don't know, a T, puts an A again. And it kind of, yes, it uses this complementarity to build this RNA strand. Or for example, when we are kind of making this RNA messenger, or to duplicate the DNA when it's kind of the cell is dividing. So yes, this complementarity of strands is used as a template for then for the protein to see and okay, it's kind of a factory, like, oh, here there is a C, here there is a G, and make the process.
Excellent. Shane, so we've got a question saying, does this dancing also happen in bacteria with a closed loop of DNA? I guess you've
Yes.
kind of shown that. You were showing these coils,
Yes.
weren't you?
So this dancing, it happens to our DNA as well. Even if our DNA is really wrapped around, sometimes what happens in these promoters, in these beginnings of genes, is that these proteins that wrap DNA disappear. So then the DNA can make this loop, and then it can make the bubble, and then it vibrates. But also, yeah, so the people need to take the image that all the molecules are vibrating all the time in our cells and in constant movement, vibrating. All the proteins are the sugars, the fatty molecules, the proteins, all everything. Yeah.
Yeah. Speaking of the fact that it's a temperature dependent thing, that where it dances, I guess it will dance more if it's at a higher temperature. Brilliant question. I love this question. If that's true, does that mean extremophiles will have a different process to DNA replication and protein synthesis than warm-blooded creatures? Is that
It's like this. It's a super question. So organisms that live in high temperatures, they are known to have more CGs than ATs. The CGs is because they are maintained together by 3 hydrogen bonds, which is a chemical interaction, and the ATs are maintained by 2 hydrogen bonds. So of course the ATs are sequences much more weaker to break, easier to break. So there is where the bubbles are formed, and CG are much more resilient. So this is one difference. These organisms have more CGs, and
yeah.
the other this difference is that they are maintaining a less level of supercooling. They don't need that. They have the temperature, the energy that it already separates the both strands of the DNA. So the under twisting is not so necessary. Yes, it's a super
Wow.
question. Yeah.
That's such a good question and a great answer as well. I love that.
Yeah.
Do the DNA bubbles that are formed reseal after some time, or is it an irreversible process? The bubble
Yeah,
forming.
they can reseal. Yeah, they can reseal. Yes.
Yeah.
Yes. So they reseal, I guess. Well, because no, they reseal because the energy is quite flat, as we said, and also if the circumstances change around them, they also reseal. For example, let's imagine that we have a DNA that it makes a bubble here, but then this stress is released because I don't know, there is this activity that overtwists the DNA, so then the DNA is correct again. Yes, it's reversible.
Yeah, I guess is that like do the bubbles ever travel along and join each other? Is that
They can do, yes, they can do.
I'm thinking this like solid state physics when you get dislocations in crystals, like it could bring back similar
Yeah,
ideas.
and
That's
actually,
interesting.
some people have used these sometimes these models as an inspiration sometimes to describe DNA. Yeah.
Yeah, yeah. There's a couple of others. I'm not sure that again this might not be in your layer of expertise, but are there vitamins and proteins that help maintaining healthy DNA? Is there anything that people can do to be healthier? I guess I mean,
Okay. So yes. So depending of what. Okay. Let me see if I'm not saying. I mean, this is in the back of my mind, but I'm not an expertise on that. But yes, depending of what we eat and what we do, then so what happens is that if for example, if we have stress, we create chemical reactions of the smoke. We create chemical reactions that chemical molecules that can react with the DNA and then break it. And for example, create mutations. So yes, so I don't know, not smoking, being not too stressed, these type of things helps the DNA to not be mutated. Yes.
excellent. Um, yeah, is the DNA on a slow metabolism person not dancing as much? I think that's
Okay, slow metabolism person. Okay, let's think about that. Okay, so here what we have. So this slow metabolism person, it's like this because of different reasons. I'm just thinking loudly now, because we are in so as an organisms we are kept in the same temperature. So we are regulated to be at 36 degrees, and everybody, as far as I know, it's maintaining the same temperature. So then the DNA is moving in the same way. I think that this person might be less having less metabolism, slow metabolism, because maybe for any reason, for a particular reason, is building less proteins. So if there is less proteins, the probability that the reaction happens is less. So that probably is the reason, but I'm not an expert on that.
yeah. A couple of interesting ones. Is over twisting or under twisting more common? Is that
So yes, so okay, so for in general, undertwisting is more common because it facilitates separation of the both strands. If you overtwist, you can see that it's very difficult then to read the genetic information. But of course, if we kind of consider extra, bacteria that live in high temperature, then this might change. Yes.
excellent? And how many bubbles are there in a stretch of DNA? Are there a few or
Okay.
hundreds or thousands or billions or
Yeah. So this is a good question. So people have tried to calculate that. Let me think. So, for example, in the level of stress that is in the cell, we might see a bubble every 1,000 base pairs, let's say. So if I don't know, I'm very bad in maths, but if we have like 3 billions of base pairs, then we might have millions of bubbles, if I'm counting that. So, but yeah, so it's more or less a thought that we might have a bubble every a kilobase pair of 5,000, sorry, 1,000 base pairs of 5,000 base pairs, something like that. So we should have lots of bubbles, and also because they are necessary. So we need to that millions of genes or thousands of genes are acting at the same time. Are kind of being read because we are living and we need to their functions. Yeah.
yeah. So with the formation of bubbles, is there opportunity for DNA strands to form hydrogen bonds with other bubbles? So I guess this is about the fact that there's now sort of, I guess there's free bonds in the bubble area. Have you observed strands that are complementary enough to permanently deform the DNA strands?
Okay, yes, this is a very good question. This is a very good question. Let's think. Let me think one moment so I'm not saying something really silly. So I would say, yeah, so, okay. So what happened is, okay, let's me here. I'm going to try to do, oh, okay, it's working. That's brilliant. I love this. Okay, so what happens sometimes is these cruciform forms. So, this would be a very super big bubble that actually the DNA is interacting with itself, and one strand is interacting with itself, the other strand is interacting with itself. And this is important in some process of recombination where okay, I not have this information in my mind, but this is when, for example, we want to, so we have two DNA strands. Let's imagine that we have two DNA strands that are relatively similar, and then we create these cruciform forms. We cut one strand of the DNA and we mix it. And this is a process that virus wants to do to integrate their genomes in cells, in bacteria, in us. And also we do in the process called meiosis to create more genetic variability. So yes, these bubbles also are very important in this recombination.
So I've got this another one that's just could visualizing DNA movements inspire new designs and sequences in robotics and AI? I
Okay,
mean, that feels
yes.
very left field.
No, but this is, I think that yes, because here the idea would be, okay, can we have a sequence that makes a loop always in the same time? Sorry, always in the same way. So, or a loop that can be done in two ways. It has a bistable state. It makes a switch, and sometimes, yeah, this is a thing that I've actually been thinking for my research, actually. So, can I determine a loop that it's always folding in the same way? So, to avoid this variability, or it's folding in two ways, and depending the presence of the protein, can we switch between one state to the other? And I think that this could be used like in robotics, these type of things.
Yeah, that's really interesting. I mean, I think so. So, is this what you do? Is this is this how your group works? Is this how new ideas came up? Where's your? That sounds like a next inspiration. Is this do ideas come from external sources? How do you come up with your ideas?
Um, okay, so I don't know. So I what I want. So my inspiration is to solve the so to understand biology, and so I think that physics is a wonderful tool to understand that. So for example, yes. So if I see, for example, I so when I was told in the degree, was told things happen, right? So this it was kind of this gene is switch on because there is a loop and this happens, but then it's kind of well, why? Right? Why the energy or the process physically is making this? What is driving that? So this is what is kind of driving me. So to really understand how the system, why I don't know the DNA is behaving, that is making the loops. So that's why I kind of became
Yeah.
interested in physics. Yeah.
I think that's true for almost every physicist and probably engineer as well, that it's our lack of satisfaction with other people's answers, isn't it? It's like someone will tell you it's just because the way it is, and never a good answer. Well, I think we're coming up to pretty much half past. So, first of all, thank you so much again, Agnes, for your talk. I think it's so enlightening and so interesting, and so interesting how physics can be used in such an applicable way in fields that people may not have imagined before. So, it's great to have you here.